In this video, I'm going to explain how the resting potential is maintained in the axon of a neuron. What I've constructed here is a model of the axon. These green lines represent the phospholipid bilayer, and I've also shown a few proteins that are inserted into these membranes. So what we have here is the inside of the axon. This is our cytoplasm, and here is the extracellular tissue fluid. Okay, the proteins that I'm showing here within the membrane are this one here, which is the sodium-potassium pump. We've also got this protein here. I'm trying to show a channel protein here, and that protein is a potassium channel. It's a passive transport channel. We've also got these two channels here. This one is a sodium voltage-gated channel, and this is a potassium voltage-gated channel. And they'll become important when we talk about the action potential. Now, the other things I need to explain to you are these blobs. The blue blobs are the sodium ions, and the pink blobs are the potassium ions. And finally, the other thing I wanted to explain to you is another thing that helps to contribute to this resting potential, which are these. These are proteins. They are large organic anions. They can't move out of the cell, and they help to contribute to that overall negative charge inside the cell compared with outside. Okay, so I'm going to explain how the resting potential is maintained now. Now, this shows the sodium-potassium pump working. It pumps 3 sodium out of the axon, and you can see those sodium ions here, and they are released out of the axon, and then in exchange it pumps 2 potassium ions into the axon. Okay, so you can see they're being exchanged there. Now, so overall we're pumping more positive ions out of the axon and fewer positive ions into the axon. So this helps to make it a little bit more negative inside than outside. In addition to this, we've got this potassium channel. And what happens here is quite a lot of the potassium that's being pumped in, because it's at a higher concentration, will actually diffuse out of the axon. And again, this contributes to that resting potential, making it more negative inside than outside. And what we end up with is a difference between inside and outside of minus 70 millivolts. So that is how the resting potential is achieved.